screen. Go ahead and pull up Canvas and start your warm-up. Today is September 18th, 2020. It is Unit 3, Day 5. I will tell you that the 5th question on the warm-up, the picture does not show up. So the answer is anaphase 2. I'll type that into the chat. Okay, go ahead and start wrapping up your warm-up. Okay, so today we're talking about the importance of genetic diversity. Hopefully you all were able to attend or watch the video from yesterday's lesson in which we talked about the sources of genetic diversity. So today we're gonna talk about why it's important that those sources are a thing, several things. Before we do that, I do want to play this quick quiz. It is only, let's see, 15 questions. But this is going to allow us to start to identify some of the differences between mitosis and meiosis. So I'll share this tab. I want to share a link to the chat. We've got 19 people on, so hopefully we can get as many people in the quizzes game as possible. I just sent a link to the chat that you can click on to join our quizzes. And the code is 611842. So go ahead and jump into the game. Thank you to Kyrie and Alyssa. We've got 9 folks so far. I'd like to see us get closer to 18. Go ahead and click that link that's in the chat, or you can go to joinmyquiz.com and enter the code. Okay, let's say 30 more seconds to get into this quiz. We got 14 folks. Thank you, thank you, thank you. Okay, I'm gonna go ahead and start it. Okay, so we should be pushing for about 80%. Let's see if we can get up a little bit higher. We're just below 50 right now. Take your time, folks. Take your time.
Hey, Mr. Rudd.
Yes.
Hey, I just joined to let you know why I'm not gonna be. I'm not at home, so
Okay. Thank you. Changing my attendance. Okay, we're just waiting on a few more people to finish up. I'll give it about 2 more minutes or 3 more minutes until 12:30, and then we'll move on. Okay, so I know there are some people who are still finishing up, but I do want to go over this because we are still a little bit behind where I'd like us to be. That's okay. We'll get there. We still have some learning to do and some reviewing to do, but I still want to make sure that you all understand these questions. So the question that we struggled with the most, and the question where students took the longest as well, was actually the first question. It says, in mitosis, the two resulting cells are, and so we've got to keep in mind that mitosis produces an exact copy, two exact copies of the original cell, and they are copies of each other as well. So the answer here should be identical to the starting cell and identical to each other. So we've got to keep in mind that mitosis stands for identical twins. And I'm going to actually put a slide in our presentation here to really get you all to see that. This is how I always remembered it when I was first learning this in high school. And I would encourage you all to think through some ways to make all of these concepts a little bit easier to understand. It's even bigger. Okay, so we've got mitosis, we've got meiosis. I'll put them in different colors for you. Oops. Okay. So this is how I remembered it when I was in school as well. You've got mitosis, which has an I and a T in the word. So mitosis stands for identical twins. Meiosis has an E and an S, so that's the egg and the sperm. All right, so hopefully that starts to get you all to, hopefully you can start to remember it a little bit better using this. I'll go back to the game so we can go over that. Let's see. So the second most difficult question for us was number 9. It says most of the cells in the human body are produced by, this should be mitosis, okay? So meiosis is only used for the egg and the sperm. We only make eggs and sperms using meiosis. Mitosis makes all the other trillions of cells in your body. All right, so mitosis is by far the producer of all the other cells, of the most cells. Then we had number 4. After meiosis is complete, the new cells, are all different. Okay, so every egg and every sperm is different, even though males have, with males have millions of cells in their bodies, millions of sperm cells in their body. Each one is a little bit different than the other. All right, so the results of meiosis are 4 totally unique cells. The process depicted here is mitosis. I can tell because, let's see, can I make the image any bigger? No. I can tell that it's mitosis because we've got 2 cells, only 2 cells, and in the end, the results is only 2 cells. So mitosis produces 2 cells, 2 identical cells. Why do we need meiosis? We need meiosis to have the number of chromosomes going into each sex cell. So keep in mind, sperm and egg meet. They need to total up to 46 chromosomes, which means they each need to have 23. So they each need to have half the number. So we just need to continue to do some review over mitosis versus meiosis. And that's what we'll do today, but please do keep this in mind. Mitosis is identical twins. Meiosis is egg and sperm. All right. So in your notes, I want you to copy this Venn diagram. Please copy this Venn diagram in your notes. You've got a circle for mitosis and a circle for meiosis. Then the overlap in the middle is where you will write similarities that they share. And you're going to take the next 3 minutes to try to organize these. I'm not sharing it yet. Sorry. Okay, I'll go back. So this is the Venn diagram that you should be copying in your notes. You've got a circle for mitosis, a circle for meiosis, and the overlap is where you will write their similarities. So on the next slide, I'm going to give you 3 minutes to try to classify or organize a list of terms into each of the circles. 3 minutes. All right, so there are your terms right there on the right. Your goal is to put them in one of the circles or in the middle. You've got 3 minutes. About a minute left. One more minute. Okay. So hopefully yours looks a lot like this. Hopefully you've been able to classify these terms correctly. So mitosis is used for somatic cells, whereas meiosis is used for gametes. Somatic cells are also known as body cells, and gametes are also known as sex cells, which are your sperm and your egg. Somatic cells are diploids. They have twice the number of chromosomes, whereas your sex cells are haploids. They have only the normal half the number of chromosomes. Mitosis produces 2 daughter cells versus meiosis, which produces 4 daughter cells. Mitosis undergoes prophase, metaphase, anaphase, and telophase only once, whereas meiosis undergoes prophase, metaphase, anaphase, telophase, and then it does prophase 2, metaphase 2, anaphase, telophase 2. Okay, so it does the whole thing twice. We end up with identical twins, identical daughter cells in mitosis, and unique daughter cells in meiosis. Every single sperm and every single egg is slightly different. They both share cell division. They both have to do cytokinesis afterwards, and they can only occur after DNA replication. I didn't include that in your list of terms over here, but you should definitely include that in your Venn diagram. So I'll leave this up for about 2 minutes to give people an opportunity to make sure they have their answers correctly organized. Okay. So today we're continuing our conversation about genetic diversity. Yesterday we talked about 5 sources of genetic diversity, and today we're talking about the importance of genetic diversity. So all organisms that sexually reproduce exemplify genetic variation. They are slightly different than one another. You can't find two totally identical individuals. Even identical twins are slightly genetically different. So genetic diversity is important because it gives species the opportunity to evolve. You should be writing down what you see on this slide, these 2 bullet points. And evolution increases their chances of survival. So we've just got to keep in mind that if we were all 100% unique, if we were all 100% identical, we would not have any opportunities to tap into new sources of food. We would only be able to survive in the same habitats. We'd be competing for the same resources, for the same food, for the same mates. So genetic diversity gives us an opportunity to spread out, to occupy the entire earth, to survive in different habitats, to eat different sources of food, so that we don't have to compete, and so that each of us has an opportunity to pass on our DNA to our offspring. And that's called heredity. So even as we have evolved, as species have evolved over time and gotten more genetically diverse, we still have a lot in common. For example, most animals have eyes that allow them to see. Of course, our eyes see things differently. Some eyes are more adapted to nocturnal lifestyles. Some are able to see at different angles. Some have greater peripheral vision, but in general, an eye is a pretty essential structure that you'll find across animal life. Many plants have the color green because they have chlorophyll inside of their chloroplasts. So that's, even though they've evolved and there are many different species of plants, many of them have that in common. So there are still common structures even as we become more genetically diverse. But the overall goal is evolution. The goal is for us to be able to not have to compete, and also so that we can survive and pass on our genes. Okay, I'm not going to do that. So humans have a pretty remarkable life cycle. And we're going to watch a video that kind of explains what happens in the very first stages of life. But I want you all to think about some things that you already know about how life evolves, or I'm sorry, how life develops.
This is how a human gets made. Every one of us started out this way. We've a pretty good idea what happens over the 9 months it takes to produce a newborn. But there's this one bit that's been missing. Right at the beginning, before this animation even starts. The very first few weeks. Now armed with new ways of growing human embryos in the lab, scientists are learning what the very beginning of human development looks like. For decades, studying this crucial early time in a human embryo's development was a technical headache. Embryos are hard to get and keep alive outside the body. And it's an ethical challenge. Guidelines put together in the late 70s and 80s prevent scientists from growing embryos for longer than 14 days. Despite the hurdles, scientists do know a bit about what happens in those critical weeks, often from animal studies or rare human tissue samples. They know that sperm fertilizes egg, and one cell grows to 2, 4, 8, and so on. Around day 5 or 6, the blob starts to feature different types of cell. Scientists call this blob a blastocyst. All being well, the cells in the blastocyst begin to differentiate. 1 week in, it implants into the wall of the uterus. What happens next has been a bit of a mystery. By implanting itself into the womb's wall, The embryo basically hides, and that makes studying it in humans impossible. One way of exploring this crucial time is to study embryos donated by people who no longer need them for fertility treatment. In the last few years, several labs have developed new ways to nurture these embryos. Their techniques have allowed them to start building a picture of human development that's more detailed than ever before. Here's a day 6 embryo growing in a lab. Cells destined to become the actual fetus are tagged in green. Those that go on to form the placenta in blue. By day 8, the cells are sorting and arranging themselves. The green fetal cells have condensed together, and in red here are cells that will form the interface between the baby and the placenta. It seems these extra embryonic structures develop a little later in humans than expected from studies in other animals. Day 10. The whole thing increases in size, and bundles of cells start developing into support structures. Surprisingly, at this stage, the embryo can direct its own development with no input from the mother's tissues. By day 12, the outskirts of the embryo are preparing to bind more strongly to the wall of the uterus. After all, if this were a natural pregnancy, the embryo would be there for the next 9 months. You can even see little holes appearing, ready for the mother's blood vessels to start supplying the embryo with essential oxygen and nutrients. By now, the embryo is signaling its presence to the mother via a hormone, the one that pregnancy tests pick up, HCG, labelled here in yellow. After 2 weeks, the teams ended their experiments in line with the ethical 14-day limit. To study what happens after 14 days, researchers had to turn to different techniques. Recently, scientists have built artificial embryo-like structures from stem cells. Using these partial models, they can study things like cell signaling, or even the formation of the primitive streak, the crucial thread of cells which guides a process called gastrulation. That's the moment the embryo decides which end will become the head. Here they use human stem cells growing in an animal embryo to explore that process. But even after this early phase is over, there is still a lot of work involved in building a body. To study later phases, scientists have made and analysed high-resolution 3D atlases of human embryos and fetuses. One team found that the left and right hands don't simply mirror each other when growing their nerves. Instead, some branches take random paths in each limb. Another group saw muscles that grew in early embryos only to disappear as the fetus developed, like these. As yet, they're not sure why. It's becoming ever clearer just how important a human embryo's first few weeks are. Many scientists hope that more research will lead to a better grasp of why some pregnancies fail and how birth defects arise. Maybe even make in vitro fertilization work better. But some just want to understand exactly what happens to this tiny ball of cells in this short spark of time at the beginning of all of us.
Okay. Oops. All right. So that was good information, but it really only covered the first 2 weeks of life. So when the sperm and the egg, before they meet, they are still just gametes. They are haploid cells. They only have half the number of chromosomes that is needed for an organism to begin to develop. But the moment they meet, the moment the egg is fertilized, they become what is called a zygote. And that zygote is now a diploid. It has the exact number of chromosomes it needs to start to develop into an organism. All right, so sperm and egg by themselves are haploids. They have half the number of chromosomes. But zygotes are diploids. They have 2N. They have the exact number of chromosomes that they need to begin to develop. So from weeks 1 through 4, those eggs are developing they're dividing very rapidly, but they're communicating to each other the whole time. And they're saying, okay, well, because you're located over there, you need to start to develop into a brain. Because you're located there, you need to start to develop into a heart. So that by week 5, when we enter the embryonic stage, those diploid cells can start to become essential organs. So now we've got many thousands of cells and many hundreds of different cell types that start to become the heart, and some others that start to become the lungs, and others that start to become the eyes, and others that start to become the brain and the spinal cord. And so that takes place between weeks 5 and 9. And then after that, from week 10 forward, we're in the fetus stage. And essentially, that fetus is just growing and developing. Many of the essential structures are there, but they need to get stronger. And so, period of growth and development takes place for about 30 weeks. Science has developed to the point where we can now support fetuses at about 25 weeks. But even only about 20 years ago, babies that were born before 33, 32 weeks had very little chance of survival because we just didn't have the technology to be able to support them. When a baby is born prematurely, its lungs are not very strong. And so breathing is a big issue. But in the last 20 years, we've developed technologies that allow us to support very young, very prematurely born babies. Okay. This is something you should write down as well. This is the human life cycle. So the earliest stages, again, we'll call that as zygotes. That happens just after fertilization. That zygote will then begin to undergo a process of cell specialization. So some cells, like I said, become used for the brain, some are used for the lungs, some are used for muscles, some are used for bone. And in order to decide what job they will do, the cells are constantly communicating. Some of those cells, once they've been organized and once they've been arranged properly, we enter the embryonic stage. And during the embryonic stage, again, we start to really get into the growth and development of first the essential organs and then everything else, the limbs, the skeletal structure. And that happens because of mitosis. Then after birth, a human begins to make either egg or sperm cells. A healthy human begins to make either egg or sperm cells. That's the process of meiosis. And the meiosis results in gametes, which are those egg and sperm cells. And then eventually those egg and sperm cells can unite in the process of fertilization. Okay, so this is important. Please make sure that you have written down this cycle. Okay, so here it is explained slightly differently. The growth and development.
Go back, please. Go back. This flight. 10 seconds, please. Thank you.
I'm AJ, okay.
Trash, bro.
You can go.
Okay. All right. So here it is appearing just a bit differently. Please keep in mind that our sex cells, the sperm and the egg, they only have half the number of chromosomes. See how it just says N here? And then when they unite to create a zygote, now it has 2N. It has the full set. So meiosis is literally only used to produce sperm and egg cells. Everything else is done by mitosis. I'm not doing that. All right, so that concludes our lesson for the day. Are there any questions at this point?
Look at a fucking.
After we done with the warm-up, we can leave. I mean, not with the warm-up, the exit ticket, and we leave.
Yeah, I'm actually gonna let you guys go now. It's Friday, so I hope you enjoy your weekend. But you can just knock out the exit ticket and then get ready for fourth block.
All right, man. See you later.
Okay, have a good weekend.
Leave.